Hello, my name is Andrew Will. I am the Noxious Weed Specialist here at Grays Harbor County's Noxious Weed Control Board. And today I'll be talking to you guys about traditional and experimental control methods and their timing for riparian and upland noxious weeds. For the species that I'll be covering today, these include knotweed, purple loosestrife, yellow flag iris, Scotch broom, and Tansy ragwort. And for each of these species, I will be giving you a brief background about the noxious weed, some traditional and experimental control methods, timing for control, and some topical research articles that I have found that are relevant to these noxious weeds and their control. So jumping right into our first one, we have knotweed. This is a perennial plant that is native to Asia, and it was likely introduced to the United States during the 1800s without the essential insects and diseases that control it. So these were desired as ornamentals that eventually escaped cultivation and then eventually spread into wild places. They have roots that are also called rhizomes. They can grow as deep as 9 feet and out laterally up to 60 feet. Though these plants do die back to the ground each year with the first frost, in each spring, they do send up numerous early shoots along the whole length of these rhizomes. The rhizomes can also push their way under streets, grow into structures, and up through pavement. There is a report that a business in the United Kingdom spent over $600,000 just to replace the parking lot after it had extensive damage from these rhizomes. And one Welsh family even found the plant growing up through their living room floor as it pushed up through their floorboards. And this plant does have a high cost for control and management, and this is because any piece as small as an inch of stem material can produce a whole nother plant, and it's often found in riparian aquatic areas. So, as you can imagine, that spreads rapidly throughout those areas. This is a really cool video showing you how quickly this plant can grow. So, in the video, each second is a week in time. It's an 8 second video, so it's about 2 months, and it's a quick little cool time lapse that I thought would be interesting to show you. Alrighty. So getting into some control methods now. We're going to first start with the chemical controls. These are highly effective for knotweed. And these are the most successful because you can get lots of different coverage with these sprays. And glyphosate is a really big one here. So these are, again, the most common types of treatments. And treatments typically occur in the summer to early fall when the plants are actively growing. So one of them, again, is glyphosate. This is a non-selective herbicide. It's a group 9 EPSP synthase inhibitor. And basically what this means is that it causes the uh, amino acid production to effectively stop, which then affects protein synthesis, effectively halting the growth of the plant. Again, this is a non-selective herbicide, so anything that is sprayed with glyphosate, any plant material will die. You can also use a imazapyr. This one is a group 2 ALS inhibitor. Basically, how this works is that it inhibits the ALS enzyme, effectively disrupting essential amino acid production that are required for growth and physiological function. You can also use triclopyr products. These are group 4 synthetic auxins that are applied with a surfactant, and essentially this forces the plant to grow itself to death. You can also use stem injections. These are less common because they are much more tedious. You have to inject each stem with a dose of around 5 milliliters. Whereas with the herbicide sprays, you can get broadcast sprays and treat larger areas more effectively. But these stem injections are really great if you're working within a sensitive area or you don't have the proper permitting to work within aquatic or riparian areas. So stem injection could work for, again, those smaller infestations. And with this, you're using undiluted glyphosate, and you want to have the concentration higher than 41%. And again, it's directly into the stem. You have to treat each stem with a dose of 5 milliliters per plant stem. So a experimental knotweed control method that you can use is covering. With this method, it's not very successful. It's very labor intensive, expensive, and it can also increase knotweed dispersal. Um, so it's definitely something you can try. It's experimental. It can work best with smaller isolated patches, and you typically have to set this up and then install it. It's going to be a long-term process. So you want to make sure that the vegetation is all cleared out, so you have bare ground. You can use a high density polyethylene geomembrane cover. And you also want to make sure with this method that you're getting around the margins of the cover because the rhizomes can send out new shoots where the cover is not covering around that margin. So you need to regularly check in and treat those shoots that are popping up near the margins just outside of the cover. Next, we're going to get into some biocontrol methods here. So within its native range, knotweed species are naturally kept in check by sap-sucking psyllids, diseases and fungal pathogens, um, environmental factors such as volcanoes, and competition with other vegetation that can create suboptimal growing conditions. So again, that sap-sucking psyllid, this is native to Japan, and it feeds on the sap within the leaves and the stems. This was approved for release within the United States for biocontrol in 2020 by the USDA, and it has since been utilized in a number of different states, including here in Washington and Oregon. I also mentioned there's a pathogen called a leaf spot fungus. This one is not really as viable as the sap-sucking psyllid. They are considering using this as a inoculant, essentially as a fungal mycelium liquid culture that could be then used to kind of dose plants, the knotweed plants, with that liquid culture because it is pretty successful in its native range at treating knotweed, as you can see in that picture there. There's also goats. You can see in that picture there, goats love to eat a lot of different types of vegetation. With goats, there can be challenges though, because if it's just a wide open area, you're not going to confirm that the goats will just target the knotweed specifically. So typically you have to have them in a closed off area where you want them to be eating the vegetation. But they can be very successful in really removing lots of vegetation quickly, especially if you have lots of goats. This is a really cool method. It's called electrothermal treatments for knotweed. So this is really commonly used in the United Kingdom, but it's not approved for use in the US yet. And essentially how this works is that a device that generates a current of around 5 to 8,000 volts of electricity is in that van there. You can see in the picture. And this is essentially a power module that then passes that current through the cord that is then attached to the lance that the operator uses. And then it passes through the lance into the stem and plant material. And what's really cool is that it simultaneously kills both the above ground and below ground portions of this plant at the same time. This is the only method that does that simultaneously. Also, there is then a earth spike, which is then inserted into the ground on the other side of the infestation, and this essentially closes the electrical circuit. And how this works is that it's the electricity that passes through the above ground and below ground portions of this plant. It effectively is boiling the water within the plants, and it causes irreversible and immediate damage to both structures. And with this method, results can vary. You can have a new infestation that's just developing that would be highly affected by this, or you could have a old growth knotweed stand, and you would probably need to increase the voltage or have multiple treatments for this to be more effective. And this is just a cool video showing you how this works. All right, so you can see that the operator had to treat each stem there, and kind of similar to the stem injection, that is the most viable method to get as much of that electricity through the plant as possible. Moving on now, our next experimental knotweed control method is a method called the fence girdling technique. This is a very simple, long-term, non-chemical control method that you can use. And essentially how you set it up, it is involved with clearing out an area. Well, first selecting an area that you know you have a knotweed infestation. You're then going to then clear away any vegetation that is in that area. You know, it could be old knotweed canes or other plants. You essentially want as close to bare ground or no vegetation as possible. You then are going to place a wire fence mesh that has approximately or exactly half-inch by half-inch openings over that desired area. And how this works is that as the rhizome network sends up new shoots each spring, they're going to get forced through those half-inch by half-inch openings. And as the stems reach a certain diameter, they're going to effectively girdle themselves in that mesh. And how this is going to work is that that's going to then kill off that growth. This is going to prevent the plant from producing the above ground growth that can then send nutrients back down to the rhizomes. So effectively, you're going to be starving out that rhizome network over time. And this can take up to 5 years to completely eradicate an infestation. And similar to that covering method, you're going to really want to secure this down because there's a lot of upward force as those stems grow upwards, and that can push your fencing. It can move it out of the way. So this is definitely something that you need to be checking on regularly. It's not like you can just set it up and then leave it and then forget about it for 5 years. You're going to have to go in, check those margins of the fence to see if any knotweed is growing up around that fence because it likely will. So it is a really cool method that you can use. Another long-term method there. And this is a really neat figure that I found from a paper talking about source sink relationships with knotweed. And this is really the bread and butter for chemical control for knotweed. It really highlights the best times to treat knotweed and also why this is the best time to treat knotweed with these source sink relationships. So if you're not familiar what a source or a sink is, I'll break it down for you really quick. So a source is a structure or structures for plants, and that are the essentially providing the energy for the rest of the plant. You can think about a mature leaf that is undergoing photosynthesis. You know, it's generating those sugars to then produce energy for the rest of the plant, right? So that would be a source. It's a source of energy for the plant. Whereas in contrast, a sink is a series of a structure or series of structures that are receiving the stored energy from that source. So you can think about a tuber that is developing or fruit that is developing. So essentially, it's receiving that energy from the source to grow. And the cool thing about plants too is that they can start as a sink and then become a source. So plants can switch back and forth between these two methods, right? And that's what's happening here with knotweed. So you can see in the spring here, or excuse me, in stage one, the rhizomes are very strong sources, right? There's no above ground plant material, and they have all of that stored energy in the rhizomes. So you wouldn't want to, spraying a foliar spray on the above ground portions of this plant is not going to do anything because all the energy is down in the rhizome. So during this time, you could do a pre-emergent and see how that could help you treat that knotweed. Because once the shoots start popping up, then they can then be affected by that herbicide. Whereas in stage two, the new growth is starting to pop up from those rhizomes. The rhizome is still kind of a pretty strong source. It has lots of stored energy, but it is, starting that transition from a source to a sink. This is a great time to apply any herbicides that disrupt the metabolism of growth. Um, and then in stage three, summer is happening with maximum growth. So you can really start to see the rhizome transitioning to a sink here. So the above ground portions are the new source, essentially, and then the sink is still happening in the rhizome. So you could spray here in June and July, but the most optimal time to treat knotweed. Utilizing these source sink relationships is in stage 4. So anytime between August and November. And this is when the plants are flowering. You can see that the rhizome is a very strong sink. So if you treat it with a foliar spray, it's going to readily pull down that herbicide into the rhizomes, and you're going to get really good efficacy, really good results with that herbicide uptake. All right, so this is just another article that I found, and it just kind of highlights some really cool different control uh, findings that I thought were relevant. So increased herbicide dose does not improve knotweed control. Utilizing maximum herbicide coverage via a foliar spray with exploiting those rhizome source sink relationships that I was just talking about is the best way to get the most out of your herbicide. Knotweed rhizome networks are mostly concentrated within the first meter of the substrate. They can, like I was talking about earlier, extend pretty far, uh, down into the soil and laterally out, but they're mostly concentrated within that first meter of substrate. And knotweed is really successful at outcompeting other plants, especially early on in the season. And this is because they utilize very rapid growth development, which allows them to outcompete plants for space and resource acquisition. There's lots of many factors that can affect herbicide translocation, uptake, and efficacy in knotweed. And the biggest linkages is between those above ground and below ground tissues. Um, you can also have varying degrees of biomass that can also affect this depth and size of the rhizome network, source sink phenology, and yeah, all these combined can affect your herbicide uptake and how effective it is. Alrighty. So we're now going to move on to our next species. This is purple loosestrife. So this was introduced from Eurasia to the northeastern United States in the early 1800s. This is both intentionally brought over here, and it also spread through other means, such as horticultural purposes. So, horticulturists really like this plant. It's very showy and pretty, and they developed different cultivars in the mid 1900s for ornamental purposes, and they thought that these were sterile. But they did a series of greenhouse and field experiments and found that this plant readily hybridizes with the native and invasive. So it was just creating very fertile hybrids, which is not great. I believe this process is not done anymore, which is good, but this also helped to spread this plant. Ethnobotanically speaking, this was brought over for medicinal purposes. Immigrants may have introduced this plant for purposes such as treating dysentery, diarrhea, and various uh, uh, other like wounds or sores or skin conditions. Beekeepers really like the purple loosestrife as well. It produces very abundant flowers on their spikes, and bees really like this. It apparently has a specific flavor for their honey that they like as well. And also another method of dispersal was ship ballast contaminant. It was unintentionally contaminated in that ballast, and also wool fleece. So wetland managers were the first people to kind of notice that this plant was really becoming a problem in wetlands specifically, and it was really degrading these wetlands. So they were the first to sound this alarm. And within its native range in Europe, monocultures of this plant can form, but they are very short-lived due to biocontrol specialist insects and pathogens that keep it in check. So with the purple loosestrife, there's also the winged loosestrife, which is a native here in the United States, and they both utilize the same pollinators to pollinate themselves and reproduce. And as purple loosestrife essentially becomes more abundant and takes over this winged loosestrife, it causes pollinator competition between these two, which adds further stress to that native. This plant also really greatly negatively impacts wetland functionality as well, as it replaces broadleaf cattail as the dominant species. This leads to increased downstream eutrophication, nutrient flushes, and it also impacts the detrital communities. So the essentially detritus is the plant material that breaks down at the end of each season, it breaks into food source for different detrital feeding organisms. And with purple loosestrife, this detritus is not as long-lived or viable as the detritus of native plants. So this is really impacting the detritus community because it's not as long-lived. I believe the purple loosestrife detritus only lasts in the fall, whereas native detritus usually lasts fall through spring. And this impacts all the way up the food chain to organisms such as salmonid species that rely on that detritus. So it has a pretty big impact throughout the wetlands. Going on to some control methods. This first slide is talking about some physical methods that you can use. These are pretty time consuming and they also have high probabilities for further dispersal of the purple loosestrife. So you can do hand removals. These work best for smaller, younger plants and smaller populations that are isolated. Again, these are time consuming. This plant can also reproduce in optimal conditions from broken stems, cut stems, and stuff like that. So you are running that risk of spreading it that way as well. Um, Especially with cutting. Uh, And you want to be careful with these methods too, because if the plant is in flower or in seed specifically, you could be dispersing the plant as you are, um, removing it. There's this method called disking where you use a device such as a tractor. You pull these discs through the soil that essentially uproots the soil and um, essentially cuts up all the plant material. But again, that's not really the best since they can uh, grow back from stems, cut stems. You can also mow as well, but this can be difficult since this plant often grows in riparian aquatic conditions. So your topography may not be exactly flat. Um, and you can also be spreading propagules this way. So this is a cultural control method that can be used. It is experimental, and this is flooding. So with flooding, you're targeting the purple loosestrife seedlings, and this can be successful at a depth of greater than 12 inches of flooding. Um, this uh, this does, however, impact the native plants, and because as you can see in that picture there, the mature purple loosestrife is perfectly happy being flooded. It happens all the time and is part of their life cycle and dispersal methods. But with the flooding, you're actually impacting negatively those native plants that are trying to germinate. So this is not the best method because, again, you're only focusing those seedlings. The mature plants are doing just fine. You're impacting those native plant seeds, and you're also leading or causing further dispersal of propagules via the flooding. So chemical control is really, again, bread and butter for purple loosestrife. You can use a glyphosate solution with a non-ionic surfactant, and all of these are typically applied when the plant is actively flowering, summer to early fall. With glyphosate, you can also apply it to just the tops of the plants alone, and that can be effective. You can also use imazapyr, which is really effective after mid-bloom. And you want to make sure with these guys as well that you're using aquatically formulated herbicides, and you want to have all the proper permitting and licensing and all of that as well. Metsulfurin can also be used to actively growing plants, again with another surfactant. This one, it can only be used in certain habitat areas, so you just have to be careful when using it uh, in those areas. Triclopyr can also be used early on in the season for focusing on those seedlings, and you can also use it when it's in bloom as well. You can also combine different herbicides, such as 2,4-D and dicamba. This formulation is broadleaf specific, which was really effective because these aquatic riparian areas often have sensitive monocot species such as sedges and cattails and stuff like that. So this herbicide method, will not harm them, which is good. So again, herbicide treatment should be conducted as early as possible in order to kill the plants and prevent seed production. These plants can produce up to 2.7 million seeds per plant per growing season, so they are pretty prolific seeders. And yeah, just making sure that you treat these plants before they go to seed is best. And um, in treating these plants, you can see in the picture there, our technician Declan Foster is spraying a pretty big infestation of purple loosestrife out on the Chehalis River. And with large infestations like this, you want to have a plan, right? So a really great method that is backed in science is you treat the margins of the infestation, and then you can kind of slowly work your way back towards the middle of the infestation each year. So that is a pretty good method for treating those bigger patches. All right, so this is just showing you the purple loosestrife life cycle here. So you can see in the spring, those seedlings are popping up. This is a great time to hand pull or chemically spray those plants. In the summer, they are actively growing and flowering. This is a really great time to do a chemical control, making sure that you're preventing the plants from going to seed because they are major seeders. And then in the fall and winter, they are, in the fall, they're going to seed. This is not a good time to get in there and try to pull the plants out because you'll just be spreading the seeds. This is a good time, however, to survey if you feel comfortable with identifying the plant when it's kind of in its more dormant stage, especially in the winter when they're just kind of dead stalks. But those are kind of the different activities that you can do throughout its life cycle. And with purple loosestrife, it has a whole suite of different biocontrol species that do different things. These first two species are two different types of leaf-feeding beetles, and the research to assess biological options for purple loosestrife began in 1986 in Europe. This is the first time that biological biocontrol programs were created just to target an invasive species that was impairing the flora and fauna of an area rather than just strictly agricultural purposes, which is interesting. Support for this program was substantial from land managers who were seeing that, really damaging effects of this plant. And there has been long-term data collected from New York, which documents that these insects have increased the native diversity over time. One of the first examples of biocontrol resulting from uh, native plant recovery. So this is a really great success story with the purple loosestrife biocontrol. And these leaf-eating beetles are really abundant across North America, and they are readily feeding on the leaves. As you can see, they can really completely do some damage and really defoliate those leaves there. These two species have similar life cycles and histories, which creates a synergistic relationship between them. So this is the root feeding weevil. Only the larvae feed on the roots, as you can see in that picture there, and then the adults will generally consume the leaf material. And they only usually do this at night. So if you were to walk by this plant during the day, you wouldn't really see any biocontrol species, but you would see the damage happening to the leaves. Next is the flower feeding weevils. The adults first feed on leaves and then they feed on the flowers as they develop, which damages the developing ovaries in the flower before they open. This reduces seed output through flower bud abortion. And one egg is laid within the flower bud, and then the larvae will then eat the flower material as they develop. Weevil activity can be observed on a plant by the distinct circular opening in the flower buds that you can see in that picture there. So this is a really cool image that I found in one of the research papers that I was looking at. And this is just showing you that biocontrol takes time to kind of take footholds within an area. So starting at year zero there, you can see that's when the biocontrol species are released. And then from there on, it takes time for the continuation of generations kind of to proliferate throughout the area. And as you can see, as time goes on, there's more biocontrol species that have reproduced. And then you start to see some native plants start popping back up. And you can see all the way up to year 25 and beyond, there is still a little bit of purple loosestrife, but you can see that this is a pretty effective transformation between a purple loosestrife monoculture to getting some biodiversity back. And I also want to highlight too that with biocontrol programs, uh, not only does it take a long time for the biocontrol species to establish and start making a damage on those invasives, it also takes lots of research and time and considerations during the preliminary phases to find species that are viable for the target species. And then Also making sure that they will not be damaging or harming any other non-target species. So there's a lot of work that goes into biocontrols. But as we've learned from some of those examples in New York, this is a highly effective method. It just takes time to develop and implement. Moving on to our next species, we have yellow flag iris. This is an obligate outcrosser in the Pacific Northwest. I'll talk a bit about this more later on. It's native to Europe, Western Asia, and Northwest Africa, and first arrived in the United States in 1771, and eventually made its way up to the Northwest in 1948. All parts of this plant are poisonous to livestock and may cause skin irritation, excuse me, in humans, typically in the form of kind of like a dermatitis, essentially. There are no known cultivars of the species, though there has been many attempts to create one by horticulturalists. Yellow flag iris produces hundreds of seeds per plant each year, and flooding is thought to play a key role in the distribution of rhizome fragments and seeds. So, in its native range, this species can become dominant or weedy, and reproduction through rhizome fragmentation is favored over seed production. So this is a really big study that Dr. David Gaskin, or excuse me, Dr. John Gaskin of the USDA did here in the Pacific Northwest in 2016. And this essentially is showing us that here in the Pacific Northwest, yellow flag iris is predominantly spread via seeds. In their study, they found that there was some rhizome reproduction, but here in the Pacific Northwest, it is almost entirely seed production, which is pretty big information. Um, and I just wanted to share this slide with you. It's kind of showing some different yellow flag iris infestations from around the world. There's a researcher named Stone who had a really great kind of passage that I'd like to read to you about this. This is from 2009, and they say that pale yellow iris reproduces both vegetatively and by seed. In Poland, reproduction from vegetative fragmentation was more common than seedling establishment. In Montana, reproduction by seed was thought to be more important than vegetative reproduction. So essentially, what they're saying is that the method of reproduction used may depend on local site conditions. And then they go on to say that on the fringes of salt marshes in Ireland, shore level influenced reproductive method. On high salt marsh sites, rhizomes were long-lived and seedlings were rare, whereas at the low salt marsh sites, rhizomes were short-lived and there were considerable numbers of seedlings. So this is essentially saying that potentially location of the infestation and also the conditions within that location greatly impact which dispersal method is used, either rhizomes or seeds. And it seems to be kind of one or the other, um, not really too much of a middle ground area. So within its native range, yellow flag iris has a number of different stressors that can deter it or impact it. This plant member is toxic so it does deter a lot of grazers, but there are some insects that like to feed on it, such as the iris sawfly. Its larvae can cause significant damage by defoliating the plant. There's also the iris borer. These caterpillars tunnel into the leaves and feed on the rhizomes, causing severe structural damage. Seed weevils and flea beetles. There's a number of different species of these insects that can feed on the seeds and foliage, helping to limit the plant's reproductive success. There's also different Lepidoptera species, approximately 10 species of moths and butterflies that can eat this plant essentially. There's also some different pathogens, including the iris root rot. This one gets onto the rhizomes and causes the leaves to turn yellow prematurely. There's also an iris rust. This is a fungal pathogen that can infect the plant. Um, Although the plant, yellow iris, does have some resistance to this. Also, some native plants can also pose a competitive nature with this plant. Different willow species and also specifically red osier dogwood. So just highlighting here the yellow flag iris life cycle. So the seeds are dispersed on water, and this generally starts in the fall. So those seeds do float. They're not going to germinate in the water typically. But once the water levels drop in the spring, the seeds are going to make contact with soil, and then they will germinate. Different water inundation and salinity levels can impact germination rates. Oh, excuse me. Um, Seedlings emerge and generally prioritize developing strong root systems first. They are growing in areas where there's fluctuating water levels, so they will develop those root systems first. The plants take generally about 3 years of growing before they can become mature and produce flowers and seeds. And typically there is dieback to the ground each year during fall and winter. These flowers are pollinated by long-tongued flies and bumblebees, and one mature plant produces anywhere from 150 to 600 seeds per season. So, getting into some control methods for yellow flag iris, specifically manual and mechanical methods here. Some small infestations of yellow flag iris can be best controlled by being pulled or dug out. You do want to make sure that you're wearing appropriate PPE for this, since they do have the toxic resin. Um, and since we know that the seeds are the main uh, dispersal method here in the Pacific Northwest, really making sure that the seed pods are removed is a big step to preventing that dispersal. So in terms of mechanical, you can mow, but again, kind of similar to the purple loosestrife, there can be challenges with that in terms of topography and stuff. You can use covering with yellow flag iris. You will likely need to get permitting if you're covering a large area, since you're covering up shoreline here. You can use different pond liners and stuff like that, using gas exchange inhibition to kill the plants, or also like blocking out the sunlight to kill the plants as well. And these, again, you need to bury it and monitor the edges of the tarp. Some chemical control methods here. Um, so you definitely need to make sure you're using aquatically formulated herbicides and having all the proper permitting and licensing. Glyphosates. So with this plant, it is a monocot, which means that only non-selective herbicides will work on this plant. So that's why you want to use something such as like glyphosates. Um, you can use a foliar spray, stem injection, which we kind of talked about with knotweed. Cut stump method. We'll talk a little bit more about that when we get to Scotch broom. This is Oh, we can also use wick whipping too. You can see in that picture there, there's an example of that. So you get an absorbent material such as a sponge or a cloth, and then you use tongs, and you can wipe the herbicide directly onto the plant there. And both the cut stump and stem injection and wick whipping, that's all really good for working in sensitive areas where maybe a foliar spray would not make sense. Maybe you have lots of sedges and cattails and stuff like that. So um, again, these methods also work really well in smaller infestations as well. There's also imazapyr. You can use a foliar spray for that. And fall treatments seem to be the more effective than spring and summer treatments here for chemical control. And this is just a table showing you some of the different concentrations and chemicals and mixes that you can use for chemical control of yellow flag iris. Note that the stem injection with glyphosate is 100% undiluted herbicide, and that's because you really want that strong herbicide directly injected into the stem. But yeah, those are some of just the different rates here. You can see foliar sprays are generally lower than other methods here. All right. In terms of biocontrol for yellow flag iris, there isn't any really approved for use in the United States, although there are different programs, such as in New Zealand and South Africa, where they are contributing momentum towards these biocontrol species. Um, there is the flea beetle, which has a really high reproduction rate, and they found that during a 3-month study that the number of captive flea beetles increased 5 times when reared on yellow flag iris. This is really good news because we know that having mass rearing efforts is great for establishing successive generations for biocontrols, especially if it's having really strong effects when reared on yellow flag iris itself. There's also this little guy called the iris seed weevil, and there's also lastly this leaf spot fungus. Was isolated from leaf spots on yellow flag iris. Um, it's, however, doesn't really have any strong impacts on yellow flag iris, as we know that they can have resistances to these types of fungus and stuff like that. All right, moving on to Scotch broom. So Scotch broom is native to Europe and was brought here to the U.S. in the 1800s as an ornamental via horticulturalists. It also is planted intentionally along some roadways to prevent erosion. It has different ethnobotanical uses, which is interesting. With the stems, you can use them to make brooms. It's also been used for infusing beer with different bitter flavors and flavorings. You can also use it in tanning leather. This plant is a really aggressive invader of early successional coastal Doug fir forests, especially here in the Pacific Northwest, and can cause a lot of negative impacts to those immediate plant communities. So, in its native range, also in terms of different biocontrol methods that you can use, there's lots of methods here, lots of different species. So starting off with the Scotch broom seed beetle, the larvae of this insect feed on and destroy a high percentage of seeds within the seed pods. There's also the Scotch broom seed weevil. It's pretty similar to the seed beetle. The larvae of the weevil consume the developing seeds, reducing the number of viable seeds in the seed bank. There's also this Scotch broom twig miner moth. The larvae of this moth mine the stems and twigs, which can weaken or kill plants' tips, reducing its overall growth and ability to produce flowers and seeds. You can see there in that picture, that's the gall of the gall mite that is formed. And these mites attack the newly forming flowers and foliage, creating those galls that reduces the plant's ability to produce seed pods. And similar to the knotweed, there's also a broom psyllid. These guys like to feed on the sap and have been used as biocontrol to help suppress populations. Just highlighting the Scotch from life cycle here. So looking first at germination, an interesting thing here is that Scotch broom seeds can germinate at a depth up to 2.4 inches. So anything deeper than that, they will have reduced germination rates, which is interesting. The seedlings can tolerate up to even 90% shade, which is interesting. Um, and they grow best in finer substrates than coarser ones. In terms of growth, they grow the most between May and July, and they can grow up to 8 feet within the first 2 years and may live 20 plus years. Scotch broom utilizes a method called asexual regeneration. And you can see in that picture, it's kind of hard to see, but that's a Scotch broom plant that we cut stump. And then you can see that new growth came back even after we cut it and treated it with herbicide. So that's a type of regeneration that Scotch broom can use even after being cut. In terms of maturity, seed production can begin around 2 years of age. Um, and within that time, it can be 2 to 3 years for reproduction. Um, in terms of dispersal, there's anywhere from 4,000 to 30,000 seeds, which can be dispersed per plant per season. And we know that the seed bank here is very long-lived, anywhere from 5 to 30 plus years. And some studies indicate that there may be up to 2,000 seeds per square foot of soil within these really dense infestations, which is pretty interesting. All right, so moving on to control methods for Scotch broom. These first ones are non-chemical, so manual and mechanical methods. You can dig or pull out the Scotch broom. However, this does create ideal conditions for the seed germination. We learned that it requires, uh, it kind of has a maximum seed depth of about 2.5 inches for before the germination rates are impacted. So if you're, digging or pulling out these seeds, you can then pull up seeds that are lower in the seed bank that are then now able to germinate. Um, you can also do cutting. It seems to be most effective for plants that are older and no longer green at the base, typically around 2 inches in diameter and higher seems to be the best. And a really good time to do this is when in the summer when droughts and kind of low precipitation are really impacting Scotch broom. They're already kind of under stress. You can also mow. This is really great if you have like a very large infestation, um, and you just want to knock down all of the vegetation, prevent it from going to seed and all of that. So this is a good method to use, and then you can also then follow up with a fall herbicide once those seedlings start popping back up. All right, so getting into some control methods here. Scotch broom can be controlled using a range of different herbicides and treatment styles, starting with triclopyr. This can be used as a foliar spray, basal bark spray, or cut stump application. So you can see in that picture there, you just essentially cut the stem as close to the ground as possible, and then you get your herbicide mix. It's generally about a 50% diluted herbicide. So it's at least 25% of active ingredient. And then you're just going to paint the margins of the stem around that cambium, and that's going to then get down into the roots and kill the plants. That basal bark method there is another picture. Essentially, you're just spraying the herbicide around the bottom of the stem all the way around, and that can then get the job done. All right, so I just explained the cut stump. So glyphosate is another one that can be used again as a foliar spray, cut stump, or hack and squirt application. So this hack and squirt application, it's generally used on trees and stuff like that. But with this Scotch broom, it can get pretty tall and almost tree-like. So this could be a um, um, good application for that. And essentially, there's this really cool tool called a hypo hatchet. So you can see that it's this hatchet that is hooked up to a hose, which is then fed through a container with herbicide. And essentially, how it works is that as that herbicide is pulled up into the hatchet, and then as you swing the hatchet into the plant material, it disperses some of the herbicide. So that's a cool method that you can use as well. And again, 25% glyphosate active ingredient for Scotch broom. Cut stump method seems to work best and uses a significantly lower concentration for those foliar sprays. All right, so some post weed control methods. You can do a prescribed burning. There is some things you need to be careful with for this. So you can use a weed torch or prescribed burning. It does have some volatile oils within the plant tissues, which are highly flammable. This leads to really strong, intense fires. And there is some toxicity in the Scotch broom as well. So some people have reported, feeling like a little bit lightheaded or things like that. So just being really cautious about anytime you're burning plants, especially if there is some type of toxin present. You don't want to be burning something like poison hemlock or giant hogweed. Just being really careful with burning plants that have toxins in them. And then burning also does stimulate the seed bank. So this could be a good method to, really get more seeds to germinate so you can get through that seed bank faster. Uh, Scotchroom is a good candidate for biochar as well. And if you're not familiar with biochar, this is essentially just the method of burning plant material down to kind of like an ash or char, and then you can then use that as like a very highly nutritious additive for gardens and stuff like that, or agricultural purposes. Mulching. So since we know that the Scotch broom seeds, have a max depth of about 2.5 inches, adding more organic material on top of that can also hinder germination. You can also use revegetation methods, such as adding native plants and stuff like that, and specifically adding in trees and shrubs that can help to outshade those plants. We did learn that they have up to 90% shade can still have viable seedlings and stuff like that. They can still access sunlight, so but they can definitely add some stress to those developing plants and make things harder for them. So this was an interesting study that was done in 2022 by Slisak et al. And they looked at two sites within one that was in Oregon, one that was in Washington. And essentially, they were looking at how the soil chemical properties, microclimates, and plant community structure within Scotch broom sites that were treated. They were kind of interested in seeing how that was impacted after the Scotch broom was removed. And they used a high quality site and then also a low quality site as well. And they found that Scotch broom's negative impacts on plant communities can be exacerbated following their removal. So while the Scotch broom is there, actively growing, it's causing negative impacts, and then those impacts are then continued, and if not, you know, heightened after its removal as well, which is interesting. They found that using EDRR, which is early detection rapid response methods to kind of target the Scotch broom as soon as it gets onto an area is the best to prevent this. Essentially, the longer that a Scotch broom infestation is established within a site, the longer it's going to take for those negative impacts to reverse, essentially. They found that the low quality sites were more susceptible to invasion and also the negative impacts that they were seeing than the high quality sites. So this can help us and inform us on management decisions. Maybe we have some low quality sites that we can keep an eye on and really help to monitor. This is a really great quote, essentially saying that removal of invasive species does not always lead to recovery to pre-invasion conditions. During this study, they found that even during those 4 years of their study, that little evidence of ecosystem recovery did happen with that Scotch broom. So again, the low quality sites were more impacted, and they also think that potentially removing the biomass of the Scotch broom during the treatments could have influenced this response that they saw. So that is something to take into account with all of this. All right, so our last species that I'll be talking about today with you is Tansy ragwort. So this plant is native to Eurasia and was first reported in North America in 1913, and it was likely brought over from Europe on trade ships via its use as supplemental ballast on ships. And then once they got here to pick up their goods or whatever they were shipping over, they then took all that Tansy ragwort and then just threw it off their ship into the area they were docking. So that's likely how it got to the United States and North America. And conditions here in the Pacific Northwest are really perfect for Tansy ragwort conditions. This is a really prime habitat for them. So Tansy ragwort is toxic. It contains these pure lizidine alkaloids that, when consumed, are metabolized in the liver, and they can cause progressive and irreversible liver damage. And something to note here too is that just eating Tansy ragwort once is not going to kill you. It essentially bioaccumulates, and the more you eat it, the more damage it's doing. And that's how it works too with the livestock as well. Interestingly, sheep are less affected to completely unaffected by this toxin, so they have been used in some applications as biocontrol for Tansy ragwort. Again, yeah, bioaccumulates in the liver. Death can occur with as little ingestion of anywhere from 3 to 7% of your body weight in Tansy ragwort ingestion. And then a kind of an interesting highlight here is that the Australian dairy industry estimated a loss of $4 million per year due to impacts from this plant of Tansy ragworts. And this plant can produce anywhere up to 150,000 seeds per year per plant. So it's a uh, pretty significant seeder. This is just highlighting the Tansy ragwort life cycle. So this plant is a biennial plant, and that just means that it has kind of two phases for its life cycle. So in the first phase, which happens during the first year of its life, it'll exist in this rosette stage, which you can see there. And then during the second year, it will then undergo bolting and flowering and development of seeds. And you can see it's got the really distinct rosette, kind of very almost kale-like, very leafy. It'll have those kind of purple stems at the base, and really distinct yellow flowers. And with the seeds, they kind of have those dandelion type seeds, so they are dispersed on the wind. So you need to be really careful, and it's really ill-advised to, pull Tansy ragwort when it is in seed. You should be spreading those seeds around. So there's some really important timing and considerations for control when looking at Tansy ragworts. So once this plant starts flowering, it's the biological clock is really ticking in terms of control. So, um, essentially, any plant that is sprayed with herbicide or even pulled mechanically with flowers will still produce seeds that can then be dispersed. So essentially, if you are pulling this plant, you need to make sure that you're destroying or appropriately excuse me, appropriately removing this plant. You know, you're putting it in a bag and then taking it to an appropriate location to be dumped. Um, Reseeding with desired plants that are natives ideally is really great when you're utilizing methods that are disturbing the soil. And grasses can also help to outcompete those seedlings. So, if you have an area with a bunch of tansy and it's just kind of bare ground, you could consider, putting some grass in there or putting some native plants that can help to, um, outcompete that tansy. Specifically, perennial plants do really well since this is just a biennial plant. Um, and you can see there highlighted at the bottom is showing you this Tansy Ragwort life cycle. So you can see on the left of each of those images is the first year where it's in the rosette stage, and then the one on the right is showing you the second stage when it's going into be reproductive. So you can see from June to October, it's starting to produce those flowers. So that's the best time to um, really make sure that you prevent it from going to seed. Some mechanical and manual control techniques. So hand pulling works really well if it's already in flower. You don't want to spray this when it's in flower again because it'll still go to seed. So you really need to be hand pulling this or cutting off the flower heads and properly disposing of them. It's also easier to pull when the soil is moist. Again, you should be bagging up this plants. Again, kind of similarly with the Scotch Broom, you can burn it, but it's not really advised because it does have that toxin in there. So you're going to essentially be, um, um, you know, burning up that toxin, and then you could be potentially breathing it in. Um, so it just is best to, um, bag up these plants if you are hand pulling them. And if this plant isn't seed again, don't pull it. You could just be, I mean, you will be getting that plant uh, but you will be also dispersing the seeds. You can also mow or cut this plant, but that produces kind of it forces the plant into like this perennial stage where it just is more long-lived and hardier. So it's not really advised. Hand pulling works really well. And then kind of similar thing with cutting as well. You can remove a lot of biomass this year, but then the next year it'll be more robust since you are cutting it. And then also just a little plug here. We have here at the county noxious weed control program, we have a dumpster that we get in the summer and fall each year, and residents of Grace Harbor County can stop by and drop off any tansy that they have. We also take some other noxious weeds as well, which is really a great program and should be utilized if you have some tansy on your property. In terms of chemical control for Tansy ragwort, there's a couple different options. 2,4-D has best results when used in the spring or fall on seedlings and the rosettes. 2,4-D and dicamba can be applied to Tansy ragwort at any time up to flowering and again in the fall. 2,4-D and triclopyr, this is commonly used as crossbow and it can be applied anytime up to flowering again. So picloram is an interesting one. This is the only herbicide that can be used even when the plant is flowering and as a pre-emergent. But this one, it is a restricted use herbicide, so it is incredibly difficult to get access to and also has lots of restrictions. So it's not really used at all. But this is the only herbicide that you can use um, when the plant is flowering to prevent seed production. Metsulfurin can also be used to tansy that's actively growing before flowering, and then also aminopyrrolid. You want to combine this one with the surfactant as well. It's typically used on the rosettes. It seems to be pretty effective with that. Right, moving on to biocontrol. So there's three species here, and they were first released in the 1960s, roughly. So the most iconic is the cinnabar moth. This is the most destructive to large infestations. Um, The Ragwort flea beetle. This one works best in lower density populations, so areas where there's not as much Tansy Ragwort. It does really well here. And then there's also this Ragwort seed head fly. This one's not as effective as the other two at controlling Tansy Ragwort populations, but it does occupy areas where these other two species did not survive. So, if you have a large area with different, maybe stressors on these biocontrol species, utilizing all three of these could work really well to really actively manage a Tansy Ragwort infestation. There's also some other species that are looked at in other countries outside of the US. These include the ragwort plume moth and the root-boring moth. So these are being looked at as well in terms of biocontrol. All right, so that's all the species that I wanted to talk to you today about. I just want to kind of wrap up by talking about some of the different tools that we have in our tool belt. So first off, I wanted to mention early detection rapid response. This is also called EDRR. And essentially, using this method is really ideal for getting ahead of the invasion curve, which you can see here. So you can see that early on, once a plant is first introduced, there's this is the lowest area that's infested by the invasive plant. And also, it's going to be the cheapest to control it. And then you can see as time goes on, this becomes harder and harder. There's more area that's infested. So, really getting an early jump on these invasive plants is the best control method. It's better than any type of control method that you can use, chemical, manual, mechanical. It's just yeah, getting ahead of this plant. So once you have used EDRR to identify your uh, invasive plants, you can then utilize something called an integrated pest management plan, or IPM. And this provides a really strong framework for uh, and will allow you to be successful in controlling different noxious weed species. It also allows you to be flexible and use different adaptations for different species, because as we saw with Tansy ragwort, different species have different limitations and things we need to be mindful of. So this is a really great framework for targeting and coming up with a plan to actually uh, treat this noxious weed. So you've used EDRR to find your invasive, and then you are implementing an IPM. But if you are not following the timing and phenology of this noxious weed, you could be, doing everything right, but then you're not actually treating this invasive plant. So kind of like what we saw with Tansy ragwort, again, using that as an example. If you're spraying when this plant is in flower, you're not controlling the plant. So just understanding that timing and phenology plays a big part in controlling noxious weeds. You want to make sure that you're really researching to find the best timing, kind of similarly to with the source sink relationships in knotweed. And then lastly, you can do all of those last three things, but if you just kind of treat an area once and say, oh, I did my control, the noxious weeds are gone now. That's not usually the case. Most noxious weeds take more than one treatment. Sometimes they can take multiple years. So stewardship is really important. You need to go back to the sites that you treated. You need to be checking in and monitoring for regrowth. So once you combine all four of those methods, that you get a really strong and sound invasive plant, noxious weed control plan. Lastly, I just wanted to plug the periwinkle reporting that we have going on. So I am the sponsor for periwinkle on the monitoring list, and I'm really interested in collecting data on this plant. So if you have seen any Vinca major or Vinca minor, I'd love to hear about it. You can scan this QR code, and that'll take you to the reporting form, which you can fill out. You can also send me an email there, and I will add your periwinkle reports to the data sets. Just if you do email me, include as much information as possible, roughly the size of the infestation, which species it is. GPS coordinates work really well. So yeah, I'd love to hear about any periwinkle observations in the state of Washington. Lastly, these are my sources. Alrighty. And that's all I have for you all. Thank you so much for your attention and time checking us out. Feel free to scan any of our QR codes there. You can send us an email if you'd like to have if you have any questions. We'd love to hear from you. And thank you again for your time. Hope you have a great rest of your day.